So here I just want to give a broad review of cell signaling. So we have 5 types of cell signaling, one of which is direct signaling through gap junctions. So if we have direct signaling through gap junctions, this is where basically we have a gap junction, which is kind of a channel that spans both cells' membranes, so cell 1 and cell 2. And any ions, for example calcium, that are at high abundance in one cell can flow directly into the other cell. And the example we used for this was in muscle contractions, especially for the heart, because you want your heart to, all those muscles around the ventricle to beat together. And so there's gap junction signaling to coordinate this. Another type is contact dependent. This is where the ligand is a transmembrane protein itself. So it can't be secreted, it's stuck to the membrane of the protein, and so when it binds to the receptor of our other cell, that requires contact between the two cells, contact between the ligand and the receptor, both of which are in the membranes. So if you see a ligand that is in a membrane, as the two cells come in contact and are held together by the ligand binding to the receptor, then that is contact-dependent signaling. Another example is paracrine. So paracrine signaling is when one cell is secreting, so making a ligand, and through vesicles going through the ER, Golgi out and released out into the vesicles, you now have ligand being secreted. And we know that in order to get a response from a signal, a cell needs to have the receptor. All right, so there's the cell's receptor, we've got ligands being secreted by one cell, it's activating another cell, that's paracrine. If the cell that's making the ligand also has a receptor, then this is autocrine, because now those ligands can activate the nearby cells, our cell 2, and can activate the cell itself, so that's autocrine. If the ligand comes from long distance, so the bloodstream, for example, if the ligand is flowing through the bloodstream like adrenaline or epinephrine or things like that, then as that is released from the bloodstream, that is called endocrine signaling. So there we have our 5 different types of signaling. We have direct signaling, we have contact-dependent signaling, we have paracrine and autocrine, and then we have the long distance through the bloodstream endocrine signaling. Now, no matter what though, we need a receptor, and there's a few things that have to happen. So when that ligand binds to the receptor, this is going to cause a conformation change. In the cytoplasmic side of the receptor. So now this is going to change in such a way that it's going to start to cause a signaling cascade. So in that signaling cascade, we have activation of several different things. All right, and so that might activate protein A, which activates protein B, activates protein C, and then eventually we're going to get a response. Now, a lot of this activation happens through a process called phosphorylation. So if you have your amino acid, all right, so if we take our amino acid, I'm going to just draw one here, NH2, carbon, alpha carbon, and then the carbonyl group. If we have an OH group on the side chain, this can become phosphorylated. And this changes the properties quite a bit. So if you add that phosphate group, that basically turns a protein on or off by altering the fold. Because it changes the properties of this amino acid. You change the properties, you're going to change the fold. You change the fold, you change the function. And this is used by the cell basically as an on-off switch for proteins. So we have kinases that add a phosphate and phosphatases that remove phosphate groups from proteins. This is often what's done in these signaling. So you basically activate protein A by phosphorylating it, and then that now is active and can phosphorylate B, and you get this kinase cascade. And this cascade is important because you can get amplification. So amplification occurs by the fact that, maybe one protein A gets phosphorylated, but each of those protein A's can phosphorylate 10 protein B's, and each of those protein B's can phosphorylate another 10 C's, so now we're up at 100. And if we had another kinase in here, even D, if all 100 of those C's phosphorylate 10 D's each, now we're at 1,000. So you can see how you get amplification of that signal as the signal goes through the cell, and that creates a strong positive response if the threshold is met for the ligand binding to the receptor. So that is our signaling cascade. We have ligand binding to the receptor, a conformation change in the receptor. We have a signaling cascade, often using phosphorylation to turn these proteins on and off, and this leads to amplification, and therefore you get a strong response if your threshold has been met.